All right, Dr. Bowen, you're up.
It's my pleasure to introduce Jeff Dunn today. I've known Jeff for, I was going to say many years, it's actually decades. But anyway, Jeff did not start out with the intent of becoming a doctor or even a scientist. He was a French literature major at Haverford, and that's where the spark of pursuing science struck him. Fortunately, he's not going to talk about French literature today, though I'm sure that would be a very interesting talk. From there, he went on to medical school at Temple and then did his residency out here at UW, which is where I met him. He actually practiced here in the Seattle community for many years and developed an interest in multiple sclerosis. He was one of the really founding of a group of us working at MS to develop a large comprehensive MS center in the region, which really was the predecessor for many of the current MS centers in the region, including the one at Swedish. From there, he went on to Stanford, and he's now the Lily Serafan Director of Neuroimmunology at Stanford University. He has received numerous awards for his research and teaching. There's one award, the Fisher's-Dunn Award, that's even named after him. He continues to be involved in a lot of science down there. He has over 50 peer-reviewed publications and is the principal investigator of Project BIG, which is a translational research initiative to develop better measures and markers for multiple sclerosis. He's the inaugural recipient of the Oscar Salvatierre Award, which is given for exceptional service and teaching excellence at Stanford School of Medicine. So with that, I'll turn it over to Dr. Dunn, who is going to talk to us today about EB virus and its role in MS. So thanks for joining us, Jeff.
Absolutely. Thank you for that really kind introduction. And is this showing properly?
That looks great.
Okay, great. So, yeah, you may have picked up on Dr. Bowen's introduction that I may not be blessed with natural intelligence, but there's some grit there. I'd highly recommend that book actually for all of you that are teaching and want to bring the best out of your trainees. It's called Grit, written by Duckworth. It's all about sticking to it, like Dr. Bowen and Dr. Brown and others did together in the MS Hub, the nonprofit that has formed the blueprint of what is now Swedish, but also is Stanford. If you've seen the MS Center at Swedish and you come and visit us at Stanford, you'll see many of the same elements because they derive from that same shared vision, which has been really impactful. Okay, so I'm really pleased to be able to talk about Epstein-Barr virus and multiple sclerosis today. You may know that this last year, there were two major publications in high impact journals, one in Nature and one in Science, that suggested a sort of a renewed or corroborated a suspected link between this virus and multiple sclerosis. And the implications are potentially enormous. It invites our consideration of multiple sclerosis, whether you believe it's an autoimmune pathology or not, that multiple sclerosis may well be, in most cases, a manifestation of Epstein-Barr virus infection, at least indirectly. And that, in turn, suggests that there may be treatment options that we've never previously considered. So what I'm going to do today, after reviewing these disclosures, my subconscious greatest bias would be toward biomarkers that I'm not going to be discussing, is to provide a review for you that I hope will be really helpful and interesting on these three things. First, even though this is not a virology lecture, when's the last time you studied Epstein-Barr virus in any detail? We'll do that briefly today. And then I'd like to take a look at the two major manuscripts that were published this year one of which our group did. So I'll have some insight, sort of insider scoop there to share with you. They're impactful. And then that inspires fresh new questions. And so I hope that we'll leave from the grand rounds this morning really asking ourselves new questions and find ourselves with renewed interest. All right, so to the virus first. This fellow in the bottom right is Dr. Dennis Burkitt. And this was the manuscript that he published in 1958 after he had seen a cohort of 38 patients with a sarcoma. He was seeing these patients in Uganda at Mulago Hospital. Young boys and girls would present with their teeth falling out and visible facial swelling. In this manuscript, this is the picture that Dr. Burkitt showed. It's quite prominent, obviously. He took tissue and sent it back to his colleagues at Middlesex Hospital in the UK back at that time. And those colleagues included Michael Anthony Epstein, Yvonne Barr, and Bert Ekong. And what they saw is what you can see up here in the upper right. So this is an electron microscopy of the tissue that was collected from a Burkitt's lymphoma specimen. And what it showed were viral particles. Now remember, this was published in the Lancet Journal in 1964. That's just a little bit after 10 years after the discovery of DNA. And so to think that, a viral interaction potentially could spark a malignancy or cancer was extremely exciting, still is, and was the basis for a lot of oncogene research that followed. The virus was looked at and interrogated more closely. It was found to be a double-stranded DNA virus, more specifically one of the herpes viridae belonging to the herpes family. To our knowledge, there are 9 of those that have been identified. This so-called Epstein-Barr virus, don't know why Chung didn't get credit. He was part of the work, but so it is, was the 4th of the 9. So human herpes virus 4 is what we now call eponymously the Epstein-Barr virus. Its architecture is seen in the bottom right in a view that's not completely clear, but it's a more complicated virus than one might think. It's got a protein nucleocapsid, a viral tegument, enveloped glycoproteins, 172,000 base pairs. So it's quite complex with 85 genes. And I think key to this is that it's got multiple latency states. I'll double back on that in just a minute. So this virus was discovered in association with what we now call Burkitt's lymphoma. Subsequent studies have shown that there's been associations also with other malignancies, including Hodgkin's and nasopharyngeal cancer, gastric cancer. It's been estimated by some that there might be 200,000 cases of cancer associated with Epstein-Barr virus on an annual basis. So that number may be meaningful when we talk about the public health potential treatments in the future. But Epstein-Barr, as you know, has also been associated with multiple sclerosis, and especially after an infection with mononucleosis. And there are about 125,000 cases of mono in the US per year, often in young adults or teenagers, known as the kissing disease, as you may know. And there's 1 million people, we think, with MS in the US. Again, these numbers I cite now to talk about public health implications as we move through this. But Epstein-Barr virus is associated with some diseases, but in fairness, it should be said that the overwhelming majority of Epstein-Barr virus infections do not cause cancer and do not cause multiple sclerosis, and in fact are asymptomatic or generally asymptomatic infections without significant residual. So Epstein-Barr virus, as you may already know, is one of the most common human viruses. It's ubiquitous. Seropositivity studies show 19 out of 20 humans have it. So if you did, if you checked everybody for whether they've been exposed to Epstein-Barr virus, 95% plus will have it. It tends to live in saliva and body fluids. And this pathway that you see here on the right might be how you contract an infection. So an Epstein-Barr virus is expressed in body fluids like saliva, hence kissing disease. Through the nasopharynx, it may infect the epithelial cells there in the posterior pharynx. Once infected, those epithelial cells will alert a naive B cell, which is then activated and picks up the virus, going through a various lytic infection plasma B cell state, processed through lymphoid tissue, which we'll review momentarily, and then ultimately becoming a latent infected memory B cell. So to review, Epstein-Barr is around everywhere. Its infection is via epithelial cells and B cells over a latency of 4 to 7 weeks. Most people are infected in childhood and don't even know they've been infected. But once infected, Epstein-Barr remains latent in human memory B cells and for life. The virus itself sort of tucks away itself in an episome within the nucleus of the B cell. So mostly asymptomatic, but chronically latent. These are, unique, well, not completely unique, but important characteristics of EBV. At the clinical level, in those patients, again, mostly teenagers, a classic phenotype might be a college sophomore, otherwise healthy, presents with really a significant gnarly adenopathy with a lot of exudate, as you see here in the upper right. The degree of lymphadenopathy that can follow Epstein-Barr virus causing mononucleosis can be so prominent as to be visible, as we see here, and also palpable. You can also see splenomegaly and a rash in multiple manifestations. So the virus can affect people, as in the form of mono with prominent clinical signs, but that's not what we're talking about today, at least not mono. Just as a reference, because this link between Epstein-Barr and MS has become so prominent that I suspect, especially my colleagues in neuroimmunology, are being asked by patients, should I get checked for Epstein-Barr? Is there anything we can do there? So just as a reference, there's multiple ways to assay for Epstein-Barr virus that are shown here. I think the question as a clinician might come up to you most directly as, have I ever had Epstein-Barr virus infection? And so I would just say that if that's the question, if for any reason you'd want to run those assays, the best would be a viral capsid antigen looking for IgG, which is a immunoglobulin that would peak 2 to 4 weeks after infection, and then it persists long-term, combined with an Epstein-Barr nuclear antigen assay, which is a little bit slower to appear after 2 to 4 months, but also then persists long-term. Those would be the ones that would be most sensitive if you were to address that question. A monospot, which might be the easiest thing to reach to or the most accessible, is not recommended. It actually has a very high positive and false negative rate. So it's not going to get you where you want to go. Just as a reference, and then there's a reference here from Ashley Waldron. Sorry, let me see. Here we go. Okay, let me have you look at the upper right here first. So I mentioned just a moment ago that most infections are asymptomatic. And in fact, most infections occur, we think, in early childhood. If there's exposure in someone of single digits in age, it appears the viral load may be lower. There's multiple latency states, specifically what we know of 3 latency states of Epstein-Barr in B cells. And if there's a childhood infection, let's say a 5-year-old, it seems as if the latency state that derives from that is latency 1 or 2. And almost always that becomes an asymptomatic infection, maybe after a viral exanthem and nonspecific symptoms. But if the infection occurs in young adulthood, it appears that some of the drivers may include a much higher viral load or degree of viral exposure. Young adults would be more likely to carry Epstein-Barr in latency state 3, and this can manifest as mono initially. Let me stress to you, I'm repeating myself, sorry about that part, but most EBV infections are asymptomatic. When Epstein-Barr virus infection occurs later in life, it's associated with mononucleosis. The higher B cell infection occurs when the Epstein-Barr virus, the primary infection, appears delayed, maybe into early adulthood. And at that time, the number of latently infected memory B cells can rise. enormously high, to maybe half or higher of the peripheral circulating memory B cells. That may be what drives sort of this virus or the latency of this virus past the normal checks and balances that might otherwise keep it symptomatic. And as I mentioned, Epstein-Barr can express multiple membrane proteins in different latency states. When there's a young adult infection of Epstein-Barr, you're more likely to have the virus carried in the latency 3 state. And in that case, the virus tucks itself away. It doesn't express itself on the surface of the cells in the form of eBNA1, 2, 3, Epstein-Barr, LP, and EBER. It tucks itself away more and is more likely to be intracellular, showing itself only as what's called an Epstein-Barr nuclear antigen during cell division only. So there's different ways to carry Epstein-Barr virus in latency. A young adult having a primary infection might be more inclined to carry Epstein-Barr in latency 3, which shows itself on the cell surface a little bit less and infrequently, but periodically. Okay, let me ask you to go up to this horrible drawing, which is mine, not an artist. And let me take you through this. So if there's an Epstein-Barr virus infection infecting the epithelium recognized by naive B cells that become lytic and processed through lymphoid tissue, you end up with a latently infected memory B cell. Now, looking to your right from that point, there's a branch in the road. So the overwhelming majority of these infections are asymptomatic, but there's a small subfraction of this where there's a deviation toward the onset of multiple sclerosis. That's what these 2 manuscripts show, which we'll review momentarily. I've talked with my colleagues. One of the great values of being here at Stanford is, having these world-class scientists that are immediately available for us, literally across the sidewalk in some cases, that we can review this with. And so as clinicians, we've told them, this is where we look here. This is the fork in the road. And if we can figure out why this fork in the road exists, that's the cause of multiple sclerosis, maybe in most cases. So why is it that a small subfraction of these EBV infected patients develop MS where others do not? That's an unanswered question, but that's where I think we can focus. And it means that we're also so much closer to being able to figure out the cause, at least in what might be the majority of MS cases. We'll see. I've told you about the epithelial cells and the B cells, but just to review immunology just a little bit more, when the naive B cell will pick up a virus or an antigen, it goes into a blast stage and then travels to a germinal center, ultimately evolving into plasma cells that produce antibodies. So let me show that to you now. If you look up here at the upper right, this shows the lymphoid tissue that a naive B cell might traffic through. So here's a naive B cell that, let's say, has encountered antigen. It will traffic then into the lymphoid tissue through the mantle zone and into the light zone, where it undergoes clonal expansion. There's then somatic hypermutation, so multiple DNA changes that affect both the light chain and heavy chain recognition of what will ultimately become antibodies. Sometimes that somatic mutation or the changes that take place increase the affinity. They increase the ability of the antibody produced by the DNA to recognize the antigen that's been encountered. And that's called, as you may know, affinity maturation. The recognition of the infected antigen becomes greater with higher affinity. There are other mutations, of course, which cause lesser affinity, that allow antibodies to not even recognize or recognize more poorly the infection or the antigen that's been encountered. And through lymphoid processing, those are deselected and undergo apoptosis. So you get the naive B cell traffics through, you get proliferation, somatic hypermutation, an affinity maturation, and then ultimately the production of immunoglobulin expressed on the surface of memory B cells. Looking down to the graph on the lower right, this actually tracks a little bit with the graph I've shown here of the lymphoid tissue. So a naive B cell that's circulating through the human body, as you may remember from medical school years ago, expresses immunoglobulin in terms of IgD and IgM. But then with antigen activation in lymphoblastic stage, IgD falls off and IgM is expressed. And ultimately, this matures to an IgG producing memory B cell. So that's sort of an interesting process. And it's good to know. And then obviously, memory B cells are sort of learned, rather than being the kindergartners of the immune system, they're the PhDs of the immune system. They've been through it and they know their antigen and they know how to attack it. So if a person is re-exposed to an antigen, the lag period for the immune response is much shorter. Time of peak response is shorter. The magnitude of an antibody response can be 1,000 times as high as with, a naive exposure. And the lifespan of these memory B cells can be long-lived with a marked increase in antibody affinity. And of course, this is the basis for vaccination. Okay, so let's pivot a little bit to multiple sclerosis. So from overall global observations, epidemiologic observations, it's been known that Epstein-Barr virus or seropositivity is a recognized risk factor of having multiple sclerosis. And if you look at the things that we know to be associated with MS risk, Epstein-Barr seropositivity is the largest, at least in Olson's review published here in 2017. A history of mono also is 2 times the risk. So if you have mono as often as a young adult, your risk of MS seems to double. There's other risk factors here too that don't have anything to do, at least directly, with Epstein-Barr virus infection, such as low sunlight exposure or adolescent obesity. But those might both be related to vitamin D deficiency, which can be immunoregulatory. By the way, the person that first taught me about the connection between vitamin D and multiple sclerosis was Dr. Bowen when I was a trainee, and he was a junior faculty member back in the day. So that vitamin D story has really had legs. So anyway, there's been a suspicion of a link between Epstein-Barr and multiple sclerosis for some time. And the story gets a little bit richer in the sense that we know that almost all adult patients with MS have evidence of prior Epstein-Barr virus infection. People with MS have higher EBV seropositivity than non-MS controls. EBV infected cells are seen in brain lesions. My colleague here, May Han, who also trained at the University of Washington, has seen that we see elements of Epstein-Barr in maybe 85% of MS brain lesions looked at in tissue, but almost rarely at all in non-MS. And then Alberto Asherio, who's the senior author of one of these 2 papers I'm about to discuss, did this wonderful meta-analysis back in 2007, in which he concluded that 99.5% of people with MS have evidence of Epstein-Barr seropositivity. Now, against that background, 94 to 95% of all people have evidence of Epstein-Barr positivity. So to be clear, a lot of people get Epstein-Barr, but they don't get MS. But then it seems as if having an Epstein-Barr virus infection may be necessary to develop MS as a first step, even though it's not the direct cause of MS. The possible pathways to this were reviewed by my colleagues here, Bill Robinson and Larry Steinman, both at Stanford, where they posited that Epstein-Barr virus infection does not by itself cause multiple sclerosis. You might get that question based on how much hype there's been about this literature. But again, if Epstein-Barr were the direct cause, 95% of us would have MS, and that's not what we see. So it's been posited that molecular mimicry, so something similar to, let's say what Campylobacter is to Guillain-Barré. So you get a primary infection, the immune system recognizes an antigen that may be similar to a self-antigen. There's a cross-reaction via molecular mimicry that mediates immune-mediated disease. it's been thought that that might be true for MS. But Larry Steinman especially was careful to point out there are other potential ways in which an Epstein-Barr virus infection could increase the risk of MS because it encodes an IL-10-like protein, which is a B-cell activator. It can have a T-cell effect. There's an overall increased inflammatory response, and EBV can transform B cells. So molecular mimicry historically has not been a slam dunk. Okay, so at this point, let's turn to those two manuscripts. The first of both published this last year, by the way. So the first of these was published in the journal Science. The first author is Bjornvik, and then Alberto Asherio, who's an extremely accomplished, extremely sage senior author, functioned as the senior author in this paper. Remarkable work. They had access to serum samples from the Department of Defense. In fact, 10 million samples, 10 million individuals and 62 million samples. So the story is here that in military recruits at the time of enlistment, baseline blood is obtained really primarily to screen for HIV. And then it's required that blood be drawn in active military recruits on a biennial basis, so twice a year, over time. And for the blood that's not used, it's stored and banked. And so Asherio and colleagues were able to access these blood assays over a 20-year period in the period or the epoch between 1993 and 2013. And then they looked at it for Epstein-Barr seropositivity. And here's what they found. First, 95% of the military recruits, even by the age of 20, so at the time that they presented and they had their first blood draw, had already been exposed to Epstein-Barr, were already showing immunoglobulin to Epstein-Barr virus. And that's consistent with what we know. Epstein-Barr is ubiquitous. Most people are infected in childhood, 95% prior to the age of 20. Then they were able to look at, by medical records, whether there had been new diagnosis of multiple sclerosis over that 20-year period. They identified 955 such cases of this 10 million cohort, 801 of which had paired samples that were matched to age and then to ethnic background. And those are the ones that they used. 35 of the 801 samples were initially seronegative for EBV, and 34 of the 35 people became seropositive prior to the onset of their diagnosis of MS. I know that's a lot of numbers, and it was for me too. I've had to go over this multiple times to get it, but let's just go a little deeper and this will start to make sense. This is the architecture of the paper that was published. And so if you look here in the central column, this just reviews that there were 62 million blood samples that were obtained from 10 million individuals, 955 MS patients identified on a medical basis, of which 801 had matched controls. It was reviewed as to when MS began, and then even though it was retrospective, the blood was collected prospectively. It was looked at to see if people had developed EBV seropositivity and at what time relative to their MS. On the left, this represents the sort of demographic background. And no surprise here, reflecting the military recruits themselves, 8 out of 10 patients who provided samples were men, male, most of which were very young, certainly younger than 30, the overwhelming majority. And I think unlike some of our clinical trials for MS therapies, which have been limited by a high prevalence of Caucasian subjects, there was a variability in race and ethnicity that reflected what America looks like. But not everybody in this cohort developed MS. Those who did showed a bit of a female deviation. That's understandable given that we think about 70% of MS patients are female. Younger onset, so the vast majority, again, were younger than 30, with a nice ethnic heterogeneity. So that's the demography. All right, and then from these data, several assays were done. One, assessment of Epstein-Barr status. So was there an association between Epstein-Barr virus infection and multiple sclerosis? There was a cross-check with cytomegalovirus for reasons I'll explain momentarily. And then these researchers, much to their credit, added these really eloquent extra considerations, which I'll discuss. Here are the results. This data is not intuitive, so let me take you through it. Look here in the upper left. The green line represents those patients who had been seronegative who developed multiple sclerosis. And in fact, 97% of these who had developed Epstein-Barr later developed multiple sclerosis. There were also patients who developed new onset Epstein-Barr virus infection who did not develop MS. That was 57%. If you look at it then, the hazard ratio of the differential between these two groups is 32 times. Or another way to say this is that the risk of multiple sclerosis increases by 32-fold after infection with Epstein-Barr virus infection specifically. The authors looked under a same analysis to see if this same dynamic might occur with cytomegalovirus, which is a nifty comparison because CMV, as you know, is also a herpes virus. It's also transmitted in saliva, so its pathways would be very similar. So it's a really nice match in that sense. And there was no differential seen. So this really suggested that EBV and MS are related. Okay, bear with me for a second. So there's a lot of data that are coming out in general that show associations. So if we have an association here between Epstein-Barr and multiple sclerosis, I reflexively, and I'd recommend especially to the trainees here, like if you see two things come together, really consider which way the causal arrow goes. So if you got A and B, you got to ask yourself, does A cause B, or does B cause A? Or does C cause both? Is it an epiphenomenal thing? So the presumption here is that Epstein-Barr may trigger multiple sclerosis. And even though this sounds weird to say, could it be the opposite? Could it be that multiple sclerosis triggers EBV infection? And that's a weird way to say it. So let me say it differently. Could the immunoregulatory imbalance of what multiple sclerosis is somehow increase the susceptibility to viral infection in individuals? It's not intuitive, but it's very sage to consider that. And so these researchers did just that in running a VIRA scan of a randomly selected 30 subjects, looking for overall response in those subjects to viral peptides. They looked at preclinical and early clinical MS, and specifically looked for reactivity of the wide range of viruses seen here. If there was a differential response in pre-onset samples and post-onset samples, we would see that in an orange bar. If in fact MS were causing an immunoregulatory disturbance that increased the risk of viral exposure or viral infection, we might expect to see a number of different viruses manifest on this assay, on the VIRA scan. But in fact, the only link appeared to be with Epstein-Barr, providing evidence that it's Epstein-Barr driving MS and not the other way around. So I thought that was nifty. The other thing this study did was to look at neurofilament light chain, which as an introduction manifests or represents end organ effect. Dr. Bowen asked me to just take a moment to review this briefly. And so instead of going into this data, let's take a little bit of a rabbit trail and review this. So neurofilament light chain is a cylindrical axonal protein. There's multiple of those. It comes in heavy forms, medium forms, and light forms. These are structural cylindrical proteins that really are kind of like girders. So you'll see them manifest mostly in axons. The longer the axon, the longer the neurofilament chains. And so you'll see these especially in heavily myelinated or large caliber myelinated axons, which are more prominent. This neurofilament light chain can leak in the event of axonal damage because it's cytoplasmic, and so it can leak, and now by advanced technology, it's measurable. And in fact, multiple studies have shown that neurofilament light chain in particular is increased compared to baseline in inflammatory, neurodegenerative, traumatic, and vascular disease. The author of this review, Gaetani, concludes over the last 2 decades, an increasing number of studies have shown that NFL, neurofilament light chain levels in CSF and blood are altered in CNS diseases and then can be correlated with disease characteristics. And so there's been a lot of excitement about this as a potential biomarker. But one of the problems is axonal damage is not specific to multiple sclerosis, for example. In this graph that's in the reference here, you'll see that the level of neurofilament light chain, which is measured in picograms per milliliter, so these are not robust levels showing up in the blood, is really not too dissimilar from what you might see in multiple systems atrophy. And there are higher levels seen with ALS and Jakob-Creutzfeldt disease. But the point is that neurofilament light is not specific to MS by any means. And the levels that we're measuring are really low. Again, picograms per milliliter. The advent of singular molecular array assays like Simoa has enabled these measurements. If you do this test, it's in a 1 milliliter red top tube. And not just the issue of specificity, but there's variation in these levels that can occur in non-pathologic states. So for example, NFL levels will tend to increase with age. Interestingly, NFL levels are typically 10% higher in the morning than they are in the evening on the very same day, according to LabCorp. And there's a lot of individual variation. So those are limitations. NFL, however, has been increased in clinically isolated syndrome cases. So that's showing end organ effect. We've all faced situations where in working up a patient or trying to figure out a diagnosis, we might get an antibody assay or something positive, but then a disease state is often diagnosed by evidence of end organ effect. So maybe NFL shows that end organ effect, but there are limitations there too. Ideally, if you had a biomarker of disease status, you'd want to track that over time to see where the disease is. But for example, if an optic neuritis patient presented to you and you checked NFL at baseline at the time of presentation, and then looked at them more than 60 days later, their NFL levels would drop as much as 45%, probably regression to the mean. There have been attempts to see if NFL levels could predict prognosis. Does it predict radiologically isolated syndrome, moving to clinically isolated syndrome, or CIS to MS? And those results have not been persuasive. And it hasn't been all that good in ruling out MS mimics. You'll see elevations in NMOSD just as much, sometimes more than you might see in MS. So one of my colleagues has said that NFL is the SED rate of our subspecialty. It says something, it reflects end organ impact, but it's got a lot of limitations and probably can't be used regularly for disease status assessment longitudinally. All right, so now back to the data from the Asherio paper. So if I've introduced NFL properly as a protein that shows end organ effect, these researchers showed that neurofilament light chain is on the y-axis here. So there was no difference in NFL levels before EBV infection between those who developed MS and those who did not. There was no significant differential in these levels at the time of the first EBV positivity, but there was a change where NFL levels began to rise after the infection. And so the authors conclude from this that EBV infection preceded not just the symptomatic onset of MS that was captured on a clinical level, but also the very first detectable sign of MS neuropathology, if you buy into the fact that neurofilament light chain is a marker of end organ effect. So I thought that was really, really nifty. So a fantastic paper. And as you may know, it was picked up in the press. There was a lot of enthusiasm about this. In practical terms, it was said if you're not infected with EBV, your MS risk is virtually zero. But after infection, your risk of developing MS increases 32-fold. The odds of this having occurred strictly by chance are considered less than 1 in a million. So it seems as if Epstein-Barr is necessary in most cases, maybe not 100% of cases, but necessary for the onset of multiple sclerosis. but they're still unanswered questions. And that brings us to the paper that our group did, published in Nature just a couple weeks after the Science paper, which showed that, in fact, molecular mimicry may be the driver. This is kind of a nifty correlation. So one of the nice things we've been able to do at Stanford is have our clinic and our clientele, offer human tissue that we then can work with our scientists, including Tobias Lanz, who's the primary author, and Bill Robinson, the senior author shown here. So what we did was, and I'm not sure this is in the manuscript, but let me give you the background story. We found really early cases of MS, the first clinical presentation, but in association with additional lesions. We did not look at just pure monophasic disease or very early relapsing MS. This paper is on the basis of only 9 subjects, but all of them had what appeared to be relatively aggressive onset of MS. We were very careful to make sure that we excluded MS mimics. One of the inclusion criteria of the data was that there be at least 10 white blood cells in the CSF when we did an LP. And as you know, you don't always see that in MS. Sometimes you'll see 5 white blood cells, all of which are lymphocytic or monocytic. So this looked to be inflammatory. After we obtained blood and CSF from these patients, plasmablasts and B cells were selected out by flow cytometry. By single cell sequencing, we sequenced the full length heavy and light chain VDJ regions. So if you remember that lymph node I showed you, we looked at the DNA that was driving those changes, and then created phylogenetic trees, which are shown here. This shows, if you have a mutation and then a second mutation and a third mutation, you can create these trees that are very specific, but they're kind of hard to look at. So in the upper right, you see these pretty colored wheels, and I want to dive into those a little bit more now. So there's 3 rows here that look at what the antibodies are. The first row of our 9 patients, you can count 9 circles across, was from blood. The second row is spinal fluid, and then the third was non-MS inflammatory states as a control. So that in MS patients, if you do a spinal tap, you'll see oligoclonal bands, which represent immunoglobulin. I remember training at the UW, walking out the back door of the cafeteria there out to the lake, or the Mountain Lake Cut, just after having talked to Dr. Alex Spence. And we were talking about the oligoclonal bands and what these represent. And at the time, which was early 90s, it was thought these bands might be nonsensical. There wasn't any known recognition. They didn't seem to recognize anything, at least by the technology that we could use at the time. So what Toby did was he looked at clonality. So what this means is, if it were true, for example, that the immunoglobulins you see in MS are just nonsensical, that they represent an immunoregulatory disorder and the antibodies are coming from all over the place, you might expect to see multiple antibodies that recognize a whole bunch of stuff, but there's no clonality there. There's not one family of recognition. But clonality, in this case, would imply intention, meaning the oligoclonal bands being produced are actively going for something that they have in common. And that's what was seen here. If you look at these color wheels, the broader wheels imply shared clonality. And as you can see in our MS patients, these wheels are a lot broader than, for example, we might see in inflammatory states, where there's a sort of a broad spectrum of different immunoglobulin. The immunoglobulin of CSF of these MS patients were after something in particular. So single cell sequencing of the B cell repertoires were done, analyzed, and sequenced, and CSF was highly clonal. Then these monoclonal antibodies were probed on microarrays that looked at different viruses to see what these clones were specifically recognizing. And in our cohort of 9 patients, 8 of the 9 had measurable and significant monoclonals binding with high affinity to not just Epstein-Barr virus, but a specific part of the Epstein-Barr virus called the EBNA1, the Epstein-Barr nuclear antigen type 1, which is a well-sequenced region between amino acid 365 to 425. So think of that. 8 of the 9 were specifically showing recognition of this one site of the Epstein-Barr virus. And then additional studies of probed monoclonals were looked at in human protein microarrays, which comprise 16,000 human proteins, which is about 80% of the human proteome, to see what in human tissue these monoclonal antibodies, these clonal tendencies might recognize. And what was found that there actually was a cross reaction with glial cell adhesion molecule, which is an immunoglobulin superfamily expressed in the CNS by astrocytes and oligodendrocytes. I know the data are kind of dense. The bottom line is that the antibodies in the MS patients that appeared to respond to Epstein-Barr also seem to cross-react or cross-recognize a glial cell adhesion molecule in human tissue. That's one of the great values of this study, suggesting molecular mimicry. So the overall picture that emerges from this is that an Epstein-Barr virus infection, which goes into a latent state, might induce does induce an immune response, which for reasons we don't yet fully understand, and might be driven in part by phosphorylation or some degree of additional mutation. There's some mechanisms here that are still elusive, may create antibodies that cross-react to a protein in the central nervous system. So Epstein-Barr may be necessary for the infection of MS or for the onset of MS, but not sufficient. It seems like this next step, this molecular mimicry, this cross-reaction to glial cam, may be what triggers MS. I don't want to represent to you that this is a slam dunk. As exciting as this is, as much of a step forward as this represents, there's still a lot that's uncertain. Like, not everybody with MS seems to have antibodies to glial cam, so what's with that? But I want to just repeat what sounded kind of dry and sterile at the beginning, which is when Epstein-Barr is carried in a latent state, it can be carried in 3 different forms. And in this latency 3 state, the virus seems to tuck itself away really effectively. And when it does manifest itself on the cell surface and makes itself visible, it does so as an Epstein-Barr nuclear antigen 1. And so maybe that's part of what's happening, is that when the Epstein-Barr relapses, then we get disease. And this may sound freakish. I'm kind of freaked out about it by myself. But the more you learn about the behavior of Epstein-Barr, the more I've come to believe that what we see in MS, at least the relapsing forms, actually represents the clinical manifestation of the viral behavior. So relapses might be potentially driven by relapses of the virus itself and its expression, which triggers an immune response. That explains a lot of things that we might see clinically. That's speculative, of course, at this time. Okay, so I'd said I was going to talk about the virus, did that, review the literature. Hopefully, I'm sure you were aware of those publications, but hopefully I gave you a little bit of an insider view and hopefully made some of the complex stuff of those papers a little bit more understandable. And now, just in conclusion, some questions. All right, so one is, should we check for antibodies against Epstein-Barr virus in our MS patients? Those who would say no would say they're going to be positive. 95% of people are seropositive for Epstein-Barr. So what are you going to learn? You're not going to learn anything. And how is that going to impact your treatment plan? But I heard from a pediatric colleague who said there actually might be a reason to check this in their pediatric patients, interestingly. So if you get a kid who presents with ADEM, let's say, or a MOGAD type of clinical presentation, it seems that negativity to Epstein-Barr virus antibodies in such a presentation in a pediatric neurology setting might strongly suggest against MS. So that's interesting. Maybe there's something there. Should we treat MS patients with antiviral therapies like acyclovir or valacyclovir? That question, I'm sure, is coming to you from your patients at times. I know especially the Seattle clientele is extremely well read and I suspect they've read these manuscripts, many of them. So the mechanism of action of acyclovir is a nucleoside analog DNA polymerase inhibitor. So arguing against antiviral treatment is the primary infection has already occurred. The B cell, the memory B cell is infected, and that's not going to be eradicated by antiviral therapy. There's no trials that would show benefit to this. And why give a medication if you don't have to? But there are some that have said, why not? It's safe therapy. It's even been used, for chronic suppressive therapy. And if it suppresses, viral expression, could it potentially reduce relapse risk? A trial that would do that in a scientifically sound way, a methodologic way, would probably be prohibitive because it would take such huge numbers and such long-term follow-up. So that's an ongoing challenge. And the third question is, well, should we vaccinate everybody for Epstein-Barr? Doubling back and how I started, remember Epstein-Barr is associated with mononucleosis. It seems strongly associated with multiple sclerosis and also, maybe 200,000 cases of cancer. So would vaccination be a thing? Those that would say no, might point out that as we saw profoundly in COVID, vaccination might attenuate, but doesn't prevent viral infection. There's no proof of reduced risk of MS or related cancers, obviously. There are potential complications with vaccination and probably prohibitive healthcare costs. But the mRNA vaccination, for example, developed by Moderna, is all based on knowing the sequence of what it is you want to downregulate immune recognition to. So the COVID vaccination was allowed in part because we had the sequence of the spike protein that expressed itself on the SARS-CoV-2. So it appears that in what might be a majority of cases, we actually know exactly the epitope where this molecular mimicry might be mediated, that 66 amino acid sequence of EBNA1 on Epstein-Barr virus. And so in fact, phase 1 studies have started of mRNA vaccination. Moderna's doing one study. There's others too that I'm not aware of, but phase 1 studies at this point. So pharma is setting up trials on that, interestingly, and we'll continue to follow that story, I'm sure. Does this newly elucidated pathophysiology have implications for MS treatment? Well, there have been those that have suggested so, that have favored a product they might be representing, for example, but not yet. The proper choice of immunotherapy is based on all of those different things we think about as clinicians, and this doesn't impact treatment quite yet. All right, so thanks. So it's so gauche to put a picture of myself up here, but sorry about that. But in the back here of this picture is the building of where we started this MS hub that I worked with Dr. Bowen and Dr. Brown. Dr. Smith had been with us in early days. And as I said, I'm so grateful for it because it was really the blueprint or the foundation of what became 3 significant centers in the West Coast that are treating, well over 15,000 patients collectively in a patient-centric, multidisciplinary way. And so nothing's been written about this, but I think that the creation of those centers has really been a wonderful thing. Shout out to the Athenian, because that's where I always had early Sunday morning breakfast before spending the rest of my life at Harborview during training days. And finally, in the bottom right, picture of me with my son at watching a Seahawks game where one of my favorite players, Marshawn Lynch, said, and is quoted as saying, "It's a team game, bro." And that's true for science as well. So that's it for me. Thanks very much for your attention. Happy to take questions.
So Jeff, there's a couple of questions in the chat, and the first one is from Dr. Fong, one of our MS docs, and she actually has 3 questions. But the first is, given that B cells act as antigen-presenting cells, what are your thoughts on latently infected B cells as a trigger for T cells to inflame and demyelinate the CNS? Yeah,
that's a legitimate consideration. In fact, there's been a push, like, we know that B cell therapies have been highly effective in multiple sclerosis cases, not perfect, but they would lyse B cells, and this suggests that maybe they would have an effect at the very base of where MS is coming from. One of the problems, for example, with the CD20 therapies, as you know, Jim, is that their size. They don't get into the central nervous system, and the oligoclonal bands are coming about from intrathecal antibody synthesis. So there's a sort of a renewed production of those oligoclonal bands in the CNS that B cell therapy may not be able to penetrate to completely. But the instinct, I think, is spot on. And one more point before I defer again. Back in the day, there was a lot of talk about Th1 to Th2 deviation. MS was thought to be a T cell disease largely derived from, what we knew of EAE. Buster Alvord was a prominent proponent of that. As you know, he trained both of us. But in fact, what we've discovered is that B cell therapy is really effective for MS as well. So good instincts, and I think that's got to be explored further.
Yeah. Let's see. Her next question is, has there been a link between EB virus seropositivity titers and MS? Because many of our MS patients have high titers, which could mean maybe something is different about the virus, or it could just be that their immune systems are revved up in general and they have high titers to everything.
Yeah, so I think that question, which is a really good one, was addressed. I hope I did it justice, but I would recommend doubling back on the Asherio paper, because I think that they tried to address that question as well as they could with their controls, especially with that Vera scan they did of the 30 randomly selected subjects, to really make it seem like this is Epstein-Barr that's driving the MS process, and it's not the MS process driving viral susceptibility.
Mm-hmm. Good. And then her third question is thoughts on ATA188 as a future therapy for this.
Can you tell me what that is mechanistically? I'm not.
And let's see, she goes on to say auto or allogenic T cell directed immunotherapy against a latent B cell in MS patients. And I actually don't know what ATA188 is.
Okay. I'm sorry not to know this. A bit embarrassing, but
that's the Atara.
Yeah. Okay. Yeah. I'm familiar with it. It's phase 1. There's certainly a reason for us to think that might be helpful. And I'm glad that that's advancing. It's early days for that, but yeah, we're actually one of the sites from the Atara study here, so I should have known that more.
Now, one other question in the chat says, does an MS patient who has clinical mono, are they different than one who has latent EB virus? So what happens if you have an MS patient and you give them acute mono?
I don't think, I'm interested if anyone has actually seen that, because again, with the Asherio data, what I think that they show pretty convincingly is the Epstein-Barr virus infection, which sometimes can manifest as mono, clearly precedes the onset of MS, both in its clinical manifestations and then even down at the level of neurofilament light chain. I suppose you could have a secondary mono infection later, but generally it's the mono that precedes the MS and not vice versa. I would think that with other viruses, if you were to get a secondary EBV infection, there'd be a significant risk of an MS exacerbation or disease worsening, but I don't think MS precedes EBV. I think it's the other way around.
Then we have probably time for one last question, and I had a question, and Dr. Rapovich also has one, so I'll roll them together into one question about, he's asking about variability in EB virus and geography. Are there subtypes related to geography? And my question was somewhat related to this, which is that EB virus is more common in northern latitudes and MS is more common in northern latitudes. That was one reason that they looked at EB virus in the first place. And one of the problems, though, is that if you migrate from a low, if you migrate before your age 15, or sorry, after the age 15, from a low risk to a high risk area, you carry your lifetime risk of a low risk because you grew up, say, in Southern California. So why do these people that grow up in low EB virus prevalent regions not come down with horrendous MS when they move to a high-risk EB virus geography?
So to the latter, I think they do. So I actually disagree with the precept. Now, I'm not a biostatistician or epidemiologist, and so maybe I'm speaking out of turn here, but gosh, the number of patients that we see who, for example, have moved from what would be maybe low prevalence areas, like let's say India, Southern India or something like that, come to the Bay Area, they come to Seattle for professional reasons, graduate school, whatever it may be. They're not 15, they're 30. And time after time after time, we see the onset of really bad MS in people that have never had it before, very often associated with a severe level of vitamin deficiency. When we see these bad outbreaks of adults who've in-migrated to high prevalence areas, their vitamin D levels often, baseline levels often single digits. I'm sure you have too, Jim. I've seen that so many times that I don't buy it. I think that the epidemiologic studies may be a little bit limited methodologically or something like that. But to the former point, Pavel's question, I would point out our experience with malignancy is really interesting. So, Dennis Burkitt saw these obvious cases of Burkitt's lymphoma, obviously, quite significant. But interestingly, if Epstein-Barr caused just that, you would see it everywhere, right? And in fact, you don't. The prevalence and the link between Burkitt's lymphoma to EBV was seen in Uganda, but is not seen in other places of the world. I mean, and it's Epstein-Barr virus. And then in others, the manifestations are different. That might well be because the virus is different or the pathway of infection or something. There's a lot there. So I think there is heterogeneity. I would affirm that point. And that also, I think, has to be investigated further. Really interesting stuff.
Yeah. Well, it looks like we're running out of time here. So I would like to thank you for giving a great talk. A lot of things to think about for it. For our participants, reminder that in the chat is how to claim your CME credit. And then they just put it up on the screen as well, how you can do that. So again, Jeff, thanks for a great talk today. And hopefully we'll see you in person soon.
I look forward to it. This is a great privilege. Thank you so much. I look forward to seeing you soon, all of you. Thanks.
Thanks.